Hello everyone. In this section, we are going to cover the topic Cori cycle, also called the lactic acid cycle. This is a beautiful topic because it connects the muscle and the liver in a continuous loop of energy recycling. So let's get started. I am Brinda Ronika, tutor, Tribhulam College of Nursing. Before we begin, let me tell you what we are going to achieve today. By the end of this class, you should be able to define the Cori cycle, identify where it takes place in the body, explain each step along with the enzymes involved, state the energy yield and understand its significance, and discuss the factors that influence how active this cycle is. These 5 points are your exam targets. Keep them in mind throughout. Now, the Cori cycle was discovered by Carl F. Cori and Getty T. Cori, a husband and wife team of Nobel Prize winning biochemists. Gertie Cori was the first American woman to receive the Nobel Prize in physiology or medicine, largely for this work. So when you hear Cori cycle, remember that name. Now let me give a real life picture. You are running full speed 400 meters. Your legs are burning. That burning sensation, that is lactate building up in your muscles. During intense exercise, your muscles are working so fast that there is not enough oxygen to support full aerobic respiration, so they switch to anaerobic glycolysis. They break down glucose very quickly, that produce lactate as a byproduct. Now, if that lactate keeps accumulating, the muscle becomes acidic, enzymes stop working, and you collapse. But the body is smarter than that. It has a system to deal with this, and that system is the Cori cycle. It takes that lactate, send it to the liver, turns it back into glucose, and send the glucose back to the muscle. Now we'll see the definition. The Cori cycle or lactic acid cycle is a metabolic pathway where lactate produced by anaerobic glycolysis in the muscle is transported to the liver through the blood, converted back to glucose via gluconeogenesis, and returned to the muscles to be used as fuel again. 4 events in that one sentence: lactate produced in muscle, transported via blood to liver, liver converts it to glucose, glucose returns to muscle. It is a cycle, a loop between the muscle and the liver. Okay. Why does this cycle matter? There are 3 key reasons are there. First one is it prevents acidosis in muscles by continuously removing lactate from the muscle. The second one is it allows you to increase and sustain exercise intensity because lactate is being cleared efficiently. Your muscles can keep working at a high level for longer. This is directly relevant to athletic performance and also to clinical rehabilitation. And third one is it maintains glucose levels and energy supply during stressful times, not just exercise but trauma, surgery, critical illness. Whenever the body needs rapid energy, the Cori cycle helps keep glucose available in the blood. One-line summary: the Cori cycle recycle metabolic waste into useful fuel while protecting the muscle from turning to acidic. Okay, now where does this cycle happen? It involves 3 types of cells: muscle cells, liver cells, and blood. Think of it as a relay race. The muscle runs the first leg, produces lactate. The blood runs the second leg, carries lactate to the liver. The liver runs the third leg, converts into glucose. The blood runs the fourth leg, delivers glucose back to the muscles. No single organ does this alone. It is a coordinated inter-organ pathway connected through the bloodstream, and it is most active during periods of high energy demand like intense exercise. Now we'll see the very important thing, the steps of Cori cycle. Here, lactate production in the muscle. The muscle runs anaerobic glycolysis. Glucose is broken down to pyruvate, and because there is not enough oxygen, pyruvate is converted to lactate by the enzyme lactate dehydrogenase. This also regenerates NAD+, which keeps glycolysis running. So lactate production is not just waste, it's keeping the energy system going. Next, step 2. Transport of lactate to the liver. Lactate leaves the muscle, enters the bloodstream and travels to the liver. Specific transporters called monocarboxylate transporters on liver cells bring it inside. And step 3. So here it is a gluconeogenesis in the liver. Inside the liver, lactate is converted back to pyruvate again by lactate dehydrogenase, but now working in reverse. Then gluconeogenesis kicks in. Pyruvate is converted to glucose through the bypass reactions. We covered this in later. The liver spends ATP to rebuild glucose from this lactate. And step 4 is releasing of glucose into the blood. The glucose produced in the liver is exported into the bloodstream, raising blood glucose levels, making it available to the whole body. And the last step, that is a glucose uptake by muscles and other tissues. The muscle takes up that glucose from the blood, uses it to run glycolysis again, produces more ATP, and if exercise continues, produces more lactate, and the cycle repeats again. Look at this diagram. Muscle on one side, glucose comes in, glycolysis runs, lactase come out, follow the arrow, lactate travels through the blood to the liver. In the liver, lactate becomes pyruvate. Gluconeogenesis converts it to glucose. Follow the arrow back, glucose travels through the blood to the muscles, a complete loop, muscle to liver, liver to muscle, continuously. Study this diagram and practice drawing it from memory. That alone will answer most exam questions on this topic. Now, the energy calculation. This is a very commonly asked question. In the muscle, glycolysis produces 2 ATP per glucose molecule. So, the muscle gains 2 ATP. In the liver, gluconeogenesis causes 6 ATP to convert lactate back to 1 molecule of glucose. Net result, 2 ATP gain in the muscle, 6 ATP spent in the liver. Now, loss of 4 ATP for the whole cycle. So, the Cori cycle has a net energy cost of 4 ATP. It is not energy efficient, but that is not the point. Think of it like borrowing money with interest. The muscle borrows fast energy by running anaerobically, and the liver pays it back but spends extra to do so. The body accepts this because the alternative, the muscle shutting down due to acidosis, is for worse. Clinically, remember this: in patients with severe liver disease, the liver cannot run gluconeogenesis properly. Lactate accumulates in the blood and cannot be cleared. This leads to lactic acidosis, a serious life-threatening condition you will encounter in clinical practice. Okay, how does the body control this cycle? 2 levels. First, enzyme activity. The rate limiting enzyme of glycolysis is phosphofructokinase. When the cell is low on energy, high AMP and ADP, this enzyme is activated, glycolysis speeds up, more lactate is produced, and the Cori cycle accelerates. When ATP is abundant, it slows down. And second one is hormonal control. Glucagon release when blood glucose is low, stimulate gluconeogenesis in the liver. It pushes the liver to convert more lactate into glucose, accelerating the cycle. Insulin does the opposite. It suppresses gluconeogenesis, slowing the cycle down when glucose is already sufficient. Cortisol promotes gluconeogenesis during stress. Epinephrine drives glycolysis in the muscle during fight or flight, increasing lactate output. So, glucagon and cortisol accelerate the Cori cycle. Insulin puts the brake on it. Next, Factors influencing activity. Two main factors are there. First one is exercise intensity. At low intensity, aerobic metabolism dominates, very little lactate is produced, and the Cori cycle runs at a slow baseline. As intensity increases, lactate production rises and the cycle speeds up. At very high intensities, sprinting, heavy lifting, anaerobic glycolysis dominates. Lactate floods out of the muscle and the Cori cycle runs at full capacity. There is a point called the lactate threshold. When lactate production outpasses clearance, athletes train to raise this threshold. The second one is glucose availability. When blood glucose is high after a meal, the liver suppresses gluconeogenesis and the cycle slows. When blood glucose is low during fasting or prolonged exercise, the liver urgently needs to produce glucose. Gluconeogenesis is upregulated, the liver actively takes up lactate and the cycle runs at full speed. So the cycle activity is determined by how much lactate the muscle is making and how urgently the liver needs to produce glucose. Let's close. The Cori cycle is one of the most elegant examples of cooperation between organs in human metabolism. The muscle and liver work as a team connected through the blood, recycling lactate back into glucose continuously. It causes the body 4 ATP net, but it prevents acidosis, sustains exercise, maintains blood glucose, and keeps energy flowing to all tissues. As for the nurses, this connects directly to conditions like lactic acidosis in liver failure, blood glucose management, and exercise physiology in patient rehabilitation. Okay, thank you.